Alright guys, this is one of the last labs I'm going to show you. This is intestinal absorption of nutrients. So, a little background here. So what we're going to use is something called dialysis tubing. You might have used this in Biology 1. And so dialysis tubing we use to represent like the cell membrane. So this is dialysis tubing. It's semi-permeable, which means it only allows certain things through. So what we're going to do is we're going to use this to represent the lining of the intestines. So, in order for when we break things down in the digestive system, we break big molecules down into their smaller components. That's called chemical digestion. So, carbohydrates are broken down to monosaccharides. Proteins are broken down to amino acids. And fats are broken down into glycerol and fatty acids. So, what we're going to show you here is that they have to be broken down into those smaller components, because those big components, if we left them as their bigger molecules like starches and carbohydrates and proteins, they're not going to be small enough to fit through the membranes in order to get out into the bloodstream where they're needed, all right, to be delivered to cells. So we're going to take a look at dialysis tubing to represent that, the lining basically of the intestines. So I'm going to attempt to do this. I usually have someone help me do this, but for dialysis tubing, we need to tie it off at the bottom. All right, so I'm going to take this, and you get to watch me struggle with this because I don't have someone to help me. But I'm going to try to tie a knot in the bottom of this dialysis tubing, slowly but surely. I know you guys all like to watch YouTube videos, at least my son does, but this is probably not the most exciting YouTube video that you've ever watched, so I apologize for that. Okay, so I've got a knot in the bottom of the dialysis tubing, all right, and I'm going to tie a second knot just to make sure it's nice and tight so that we don't have things leaking out of my knot, all right, because what we want is things to come out of the dialysis tubing in the pores in the dialysis tubing, not out of my knot. All right, so I think I have a knot there at the bottom. All right, so I am going to take my scissors and I'm going to cut off the excess string here. All right, so got a knot in the bottom of my dialysis tubing. So I'm going to soak this again. All right, so what we're going to add to the dialysis tubing then is some starch and some glucose. So starch is a big polysaccharide, and we're going to assume that that's not going to fit through the pores of this dialysis tubing because it's too big. And we're also going to put glucose, which is a monosaccharide, and we're going to hope that that does fit through the dialysis tubing, all right, and is able to be absorbed into the bloodstream or be absorbed out of this dialysis tubing. So if I rub this dialysis tubing between my fingers, it does open up. All right, so what I'm going to do is add about 2 milliliters of starch and 2 milliliters of glucose. Whoops, almost spilled the starch. All right, so I'm going to add, see, it adds here. So I'm going to add 2 milliliters. I'm going to add a little bit more just because I have a huge dialysis tubing. All right, so what I added there was about 4 milliliters of starch. And I am going to add about the same amount of glucose to my dialysis tubing. Right. And all right, so got starch and glucose in my dialysis tubing. So what I'm going to do now is again struggle to tie off the top, right, of my dialysis tubing. See if I can do this without spilling it. All right, hope everyone is doing okay at home. I wish I was very sad coming into my classroom today, seeing it very empty. Like I looked at all the schedules that I had posted, like there was supposed to be a dress down day, fun dress up day the week that we were supposed to come back, and I looked at all the stuff I had on my calendar, and that made me the most sad, seeing all that stuff that wasn't going to happen. So it was sort of depressing coming in here today, but at least it's better than being at home, I guess. I don't know. At least I got to do something. So there's my dialysis tubing. I'm going to cut this extra off the top since I tied it. All right, so I tied off the top of the dialysis tubing, and it looks like this. All right, so what we're going to do is we're going to let it sit in a beaker of water. All right, so I'm just going to let it sit in there for about 30 minutes, and then we're going to come back and test it. So what we're going to do is we're going to test the water in here. So this is just water right now. And at the end, I'm going to come back and test this water. We're going to see then, is there any starch in this water, which means starch came out of my dialysis tubing into the water. And is there any glucose in the water, which means glucose was able to come out of the dialysis tubing into the water. So after it sits for 30 minutes, we're going to test this water and see what's in it, using iodine to test for starch and Benedict's to test for glucose. Okay, so there it goes. We're just going to let it sit in there, all right, for 30 minutes. And I'll come back and show you part 2. All right, see you in a bit.